So the very first chapter number 1 that we are going to start today for AS level biology is cell structure. Can you tell me what are we going to study in cell structure?
Like the organelles
Good, the
and
organelles. What are organelles?
they're the specialized like things inside cells that make up the cell.
Exactly. So explain that cells are the basic units of life. Then we are going to talk about some units of measurement, microscopy, light microscopy, structure and functions of those structure. Now in IGCSE and whatever you have done, you have talked about cells, the basic organelles, and you have labeled the structures found in the cells which were organelles. For example, you have stated that we have plant cells. If this is a plant cell has a cell wall, and inside the plant cell, we have, let's say, these chloroplast and all these labeling. In A levels, you are also going to label this chloroplast and these separate organelles as well. This is going to be one addition, and it's going to be very tough as well. We'll be using temporary preparations, how to observe cells, how to draw, measure cell structures using micrographs. Then we also have magnifications, okay? And this is the same thing that you've done previously as well. What is the difference between light microscope, electron microscopes? What are the common structures found in cells in electron microscope? The role of ATP in cells. Can you tell me what is ATP?
I think it stores energy.
Exactly, it's the energy currency. Then we'll also describe the structure of bacteria and compare the structure of prokaryotic cells and eukaryotic cells. Ever heard of these two terms before?
Prokaryotic doesn't have nucleus, and
Good,
eukaryotic has.
good, exactly. For example, eukaryotes, we humans, animals, plants, many types of fungi are eukaryotes. Bacteria, for example, is a prokaryote because our DNA is enclosed inside a nucleus. It's not lying around freely in the cytoplasm. And this is a very, very important topic from your unit number 1. And then describe the structure of viruses. This is very easy. Okay? There's nothing new in viruses that you don't know still. So cells are the basic unit of life. Every structure, every tissue makes organ makes organ system. Nothing, our whole body is made up of cells. Nothing was possible without the cells. Cells are the smallest unit of the human body. Towards the middle 19th century, scientists made a fundamental breakthrough in our understanding of how life works. And they realized that the basic unit of the life is the cell. And the origins of this idea go back to the early days of microscopy when an English scientist, Robert Hooke, decided to examine thin slices of plant material. Robert Hooke was a scientist who first discovered these thin slices of cells and then came to know how does the structure appear. This was the very first structure that was discovered by Robert Hooke. This is an important question for your MCQ. And in 1665, he published a book containing this particular drawing that he saw. If you examine the drawing, you will see regular structures that the Hooke law called cells. Now, in this drawing, he saw that there are very similar structures. You can see small boxes, and they are all similar. They are all similar, and he called them cells. This was observed under a normal light microscope. Each cell appeared to be an empty box surrounded by a wall, and Hooke had observed and described without realizing it that the fundamental of all living things is actually the cell. He didn't know that he has discovered such an amazing thing that would actually turn out to be the building block of all living things. Although we now know that cells of cork are dead, Hooke and other scientists made further observations of cells in living materials. However, it was not until almost 200 years later that a general cell theory also emerged. Now, the first discovery was made in 1665, where he actually published a book containing this drawing. And after 200 years, scientists made further observation as well, and they came up with the cell theory. Cell theory is asked in the paper as well, important for paper number 2. This was done by two German scientists 200 years after Robert Hooke discovered the first cell. And in 1838, Schleiden, a botanist, suggested that all plants are also made up of cells. And then Schwann, another year later, he was a zoologist, suggested the same for the animals. So who discovered plant cells? Schleiden. Who discovered animal cells? Schwann. Got it?
Yeah.
So the question is, what are the contributions of Schwann and Schleiden in discovering? And it was then soon also realized that all cells come from pre-existing cells by the process of cell division. This raises obvious question on where the original cell came from. Now, original cell came from that you have studied in previous, that we have atoms. Okay. Atoms form what when they combine together? Can you tell me?
Molecule.
Good. Atoms formed molecules. Molecules formed what? Organelles. And organelles formed cells. Got it? Then after cells, you have what? Tissue. After tissue, you have organs. And after organs, you have organ system. Can you give me an example of an organ and its organ system?
Heart, and then circulatory system.
Okay. Another example?
The lungs and respiratory system.
Good, good. Okay. So why cells? A cell can be thought as a bag in which the chemistry of the life occurs. The activity going on inside the cell is therefore separated from the environment outside the cell. The bag or cell is surrounded by a thin membrane, and the membrane is an essential feature of all cells because it controls exchange between the cell and its environment. It can act as a barrier, but it can also control movement of materials across the membrane in both directions as well. Can you tell me what does the term partially permeable membrane mean and what does freely permeable membrane mean?
Partially permeable means it lets like smaller molecules in, but then the bigger molecules it doesn't allow. But then freely permeable, it lets everything in.
Well, it's not necessary. It's always the small molecules can pass. Even not all small molecules can pass. Should say that it's selectively permeable. It doesn't mean that only small ones can pass, which means that any harmful small substance will also go inside the cell. No. Partially permeable membrane means that it can only allow selective substances to pass through. It's selective permeable membrane, partial permeable membrane. It can also allow some large molecules to pass, and it can also stop some small molecules which are not needed in the cell or which are already in very large amounts inside the cell already. Cell wall is partially or freely permeable.
Cell wall is freely permeable.
Exactly. Cell wall doesn't have any control on what exists or enters the plant because the chemicals of the cell would simply mix with the surrounding chemicals if the partially permeable membrane was free. If it was free, all chemicals of the cell would mix, diffusion, and it would become same inside and outside as well. There would be no difference between cytoplasm and the cell. So there are two types of cells. What are those two types of cells?
Prokaryotes and eukaryotes.
Good, exactly. So during the 20th century, scientists now studied the cells of bacteria. Now, bacteria is a prokaryote. And then they studied more complex organisms as well, such as plants and animals, which are eukaryotes. And they began to realize that there were two fundamentally different kinds of cell. Some cells were very simple, but some were very much larger for the complex. The complex cells contained a nucleus surrounded by two membranes, and the genetic material DNA was in the nucleus. In the simple cells, the DNA was not surrounded by membranes, but apparently free in the cytoplasm. So the very main difference in two of the discoveries, this is the first discovery, and then this is the second discovery. In prokaryotes, the DNA was free, but in eukaryotes, it was enclosed inside a nucleus. What is DNA?
It stores the genetic material.
Genetic information about the species. ठीक है. So important definition, only cell is the important definition from this keywords. Cell is the basic unit of all living organisms and it is surrounded by a cell surface membrane and contains the genetic material DNA and cytoplasm that contains organelles. Organelle is a functionally and structurally distinct part of the cell. For example, ribosomes, mitochondria, nucleus, chloroplast, vacuole, cell wall, cell membrane. All of these are organelles that together make up the whole cell. Nucleus or nuclei plural is a relatively large organelle found in only eukaryotic cell. Why is it not in prokaryotes?
Because the DNA is outside.
Because the DNA is free in prokaryotes. It is present where in prokaryotes?
Cytoplasm.
Exactly, it's just swimming around in the cytoplasm. and it is absent in prokaryote because nucleus contains the cell DNA and therefore controls the activity of the cell. It is surrounded by two membranes which together form the nuclear envelope as well. Organisms made of cells with membrane-bound nuclei are known as eukaryotes, while the simpler cells lacking the membrane-bound nuclei are known as prokaryotes. Eukaryotes are thought to have evolved from prokaryotes more than 2 billion years ago. Prokaryotes include the most important example, which is bacteria, and eukaryotes include animal, plants, and fungi, and some other organisms. Got it? So what are the eukaryote? An organism whose cells contain nucleus and other membrane-bound organelles. What does this term mean? Other membrane-bound organelles. What are those organelles then?
Uh, like endoplasmic reticulum.
Good, exactly. Endoplasmic reticulum. Where did you study that?
In IGCSE, I think.
Endoplasmic reticulum. Was there an IGCSE? Just a minor labeling, I guess. And detail you're going to study now. Okay. Prokaryote
Oh,
is an organism whose cells do not contain a nucleus or any other membrane-bound organelle. I guess you were a student of IGCSE extended syllabus or core.
extended.
Okay, then yes, you must be knowing about rough endoplasmic reticulum. Good. Okay, got it. Cell biology and microscopy. Now, this study of cells first started by Robert Hooke, then Schwann, then Schneider, and then came German scientists and botanists and many more who have finally formed the whole structure of the cells. And it all stops when we are able to differentiate between prokaryotes and eukaryotes. The study of cells have given rise to an important branch of biology, which is known as cell biology. Cell biologists study cells using many different methods, including the use of various types of microscope. There are two fundamentally different types of microscope, light microscope and electron microscope. Now, this is very important according to your paper number 2 theory. Both use a form of radiation and you have to write down the similarities and the differences. This is the main question. They are not written over here like in a table form, I guess, but you have to make that table on your own as we are going to study. Okay, so you have to list down all the similarities and most importantly, differences between a light microscope and an electron microscope. In your old edition book, there was a table which was differentiating, but I don't think they have added a table in this new edition, so you're going to have to write it. Both use a form of radiation, a first similarity, in order to see the specimen being examined. The light microscope uses light as a source of radiation, while the electron microscope uses electrons as the radiation. So what's the similarity? Both use radiation. But light and electron, light may light radiation, electron may electron radiation. Can you tell me what does micrometer mean and millimeter mean? How do we convert it? Millimeter is what?
1 millimeter is 1,000 micrometer.
Good, exactly. So what's the conversion? How would you convert, let's say, from millimeter to micrometer?
1,000.
Good. 5 millimeter would mean 5,000 micrometer. And what is nano? 9 times. If this was 3, this was 6, this is 9. So these conversions you have to practice as well. These are very important. So millimeter is 10 to power minus 3. Micrometer would be 10 to power minus 6. And nanometer is the smallest, 10 to power minus 9. These are the symbols that we use for millimeter, micrometer, and nanometer. Before studying light and electron microscopy further, you need to become familiar with these units as well that we have seen. We call these the SI units, System International Units, that will be used all over in biology. In this system, basic unit of length is meter. So we have to convert everything in meters. If I ask you, what does 3 micrometer mean in meters? So you're going to write 3 into 10 to power minus 6 meters. If I ask you what is 9.5 nanometer converted to meter, so what would you do it? 9.5 into 10 to power minus 9 meters. Got it?
Yeah.
All you have to do is just write this. Nothing else. Okay? And then your conversions, and I'll be giving you questions right now to solve as well. It would become very, very easy. What does kilo mean then?
Uh,
Let's
what's
say
a kilo?
meter to kilometer. How would you do it?
1 kilometer is 1,000 meters.
Good. And if you have to go from meter to kilometer, you would divide by 1,000. Got it?
Yeah.
So the smallest structure visible with a human eye is 50 to 100 micrometer. Okay? Smaller than this micrometer, our human eye cannot distinguish between any two objects. If there are two lines, let's say, drawn on a wall that have a difference of 14 micrometer, you won't be able to observe it because we have a highest range of only 50 to 100 micrometer. We can observe the difference in two threads. But smaller than this, we can't see the difference and we can't distinguish that is there a gap between those threads or not. So the cells in your body vary from size about 5 micrometer to 40 micrometer, and it is difficult to imagine how small these cells are, especially when they are clearly visible using a microscope. An average bacterial cell is about 1 micrometer across, and one of the smallest structures you will study in this book is the ribosome. And ribosomes can there be, tomorrow we'll be studying two types as well, 70S ribosomes and 80S ribosomes. Plant and animal cells. Can you tell me three differences each between a plant cell and an animal cell?
Plant cell has cell wall. Animal cell doesn't have cell wall.
Good.
Plant cell has a large vacuole. Animal cells have small vacuoles.
Good.
And plant cells have chloroplast and animal cells don't have chloroplast.
Amazing. Good. Okay. So when we observe plant and animal cells with a light microscope, that diagram you already know, and I'll be giving you right now a question to draw those as well again. Microscopes that use light as a source of radiation are called light microscopes. Figure 1.3 shows how a light microscope works. This is a simple light microscope. It uses light as a radiation. We have an eyepiece lens, an objective lens, condenser lens, and you don't have to remember like the diagram. There is nothing that you have to do with the diagram. We are only going to study what's the difference between a light microscope and a electron microscope. So what are the different structures that you can observe in this diagram? Tell me.
Nucleus, and then cell membrane, cytoplasm, mitochondria, ribosomes.
Hmm.
Uh, the Golgi apparatus is
Golgi
new. Like
apparatus. Yes,
I don't
exactly.
know
It's new.
what's
Exactly. It's gonna be new. Nucleolus, chromatin, nuclear envelope, and all of these. Centriole is also going to be new. Centriole will be studying in chapter number 5, the mitotic cell division chapter. Okay, it's gonna be there in chapter 5. Golgi apparatus, we'll be studying it in detail in chapter number 6. Mitochondria in detail, you're going to study that in chapter 12, A2. A2 का first chapter, chapter number 12 is mitochondria. Cell surface membrane we're going to study in AS chapter number 4. Okay, so all of these have even literally separate chapters. So this was just a structure and a generalized drawing of an animal cell that you have to memorize, and you'll be drawing it right now as well. Then we have a generalized plant cell as well, and both of these are seen with a light microscope. All structures can be easily seen. We call the diagram a photomicrograph. Photomicrograph is very important in your ASP3. P3 is your practical exam. In your practical exam, you will be given a microscope through which you have to see whatever the CIE examiner is going to give you. And whatever you see, you have to draw a basic structure of that as well, whatever you see, and then the examiner marks you. P1 is your MCQ, P2 is going to be your theory, and P3 is going to be your practical exam. Got it?
yeah.
We'll be doing P3. Obviously, we can't demonstrate it. Okay? We are not taking physical classes, but I teach P3 from the demonstration videos. I'll solve the papers as well. I have those solved solution, the tables, the notes, the precautions, how to write the method, how to perform experiment, and physically, I'll be showing you on video how we are going to perform that. So in the last 2 months, we'll focus on your P3. It's not that big of a deal. So figure 1.6 shows some human cells as well, and a plant cell that was taken from leaf. And they also show how they were magnified 400 times more. For example, this. Now, what are the new structures that weren't there in the animal cells above? Can you tell me? What are the extra structures that you see over here?
Plasmodesma,
Hmm.
chloroplast, granule,
Yeah.
the large vacuole,
Hmm.
cell wall, lamella.
Good. And this is a very, very important diagram. Middle lamella is a layer that holds different plant cells together. For example, you know the structure of a plant cell. We have palisade mesophyll cells, right, which are responsible for performing photosynthesis, and they are all lined up closely together, packed. So when they are closely packed together, they have a thin layer in between them that joins them. That is known as middle lamella. And the tunnel for the exchange of substances from cell 1, this is cell 1 and this is, let's say, cell 2, that is known as plasmodesmata. We'll be doing this in your chapter number 7, AS biology, which is transport in plants as well. Then we have a tonoplast. Tonoplast is just the name of the vacuole membrane, and the cell membrane is known as cell surface membrane. Remember, you're not going to use the term cell membrane. You always have to use cell surface membrane, which is next layer to the cell wall. The outermost layer in an animal cell is the cell membrane, and cell membrane is the second outermost layer in plant cells because the first outermost cell layer is the plant cells. Then we have the chloroplast, and inside the chloroplast we have these granules that contain those thylakoid membrane sacs as well. You are going to do this in detail in your chapter 13 A2 biology next year 2026. Then we have Golgi apparatus, nucleolus. Nucleus contains two things. The darkest structure of the cell is known as nucleolus. Can you tell me what is the function of nucleolus?
Uh, is it the cytoplasm of the nucleus?
No, that is known as nucleoplasm.
Oh, okay.
Nucleolus.
Uh,
Yeah.
I don't know.
Nucleus is actually the boss, the head of the cell. You have studied that it was the nucleus, but inside the nucleus also has an head office, the main controller, which is the nucleolus. Nucleolus is present in the nucleoplasm, and nucleoplasm have these chromatins as well. So don't worry, we haven't studied any of these. This is just a diagram. These are all going to be studied in detail when we start the each and every organelle. Let's look at this: cytoplasm, mitochondria, Golgi. All of these we'll be doing in detail. Okay, this is just an overview. What do you think? What is the structure that you can see in the photo micrograph?
Uh, cell wall and chloroplast.
So these are plant cells. Looks like, okay. What about these?
That looks like an animal cell.
Animal cell, exactly. Let's see. These are the cells of a lining of a human cheek. You can see the nucleus. Nucleus is easily available, okay, and it can be easily seen as well. Then these are taken from a moss leaf. We can identify it by the many green chloroplasts that are also visible inside each cell. And the grana are just visible as black grains. Okay? Remember, light microscope gives you less magnification. Electron microscope gives you a many magnification, a huge magnification, and plus a higher resolution as well. What can you tell me? What's the difference between magnification and resolution?
Uh, I'm not sure.
Magnification means how enlarged the object is, right? This is known as magnification. Simple.
And
Resolution.
then it's
Mhm.
like how resolution is like how clear the image is.
Exactly. ठीक है. You close. Focus. When we use a mobile phone camera, what do? You try to focus, right? What is that? Resolution. Let's say in a light microscope, you can just see this, a small line. But when you look at it in the electron microscope, it is something like this. What do you understand now? What is resolution? In light microscope, you just saw a line, but this is how it looks in electron microscope. Resolution is the ability to distinguish between two images. Got it? Which we also call as
Yeah.
focus. Focusing means what? Making the image, what do you call that? Sharp. When we make an image sharp, sharp edges, okay, it should be focused, HD. So it is very therefore easy to see when we're looking down a microscope that a deeply staining material, the deeply stained material would obviously be the nucleus. Inside its there are chromatins. When we talk about a structure of a nucleus contains nucleolus, then it also contains chromosomes. And if the chromosomes are tightly packed like this, they would be called as chromatin. So this is known as chromatin. These loosely packed, this is also the DNA, chromatin is also the DNA. Only the coiled and loosely structured are known as chromosomes. Chromosomes contain DNA. Nucleus contains chromosomes and chromatin. They contain DNA. Then the cytoplasm or nucleus is known as nucleoplasm, and the darkest structure of the nucleus is known as nucleolus. The membrane of the nucleus would be known as nuclear membrane. Nuclear membrane. And any pores or channels that are there in the nuclear membrane, they are known as nuclear pores. So what's the labeling? Chromatin, chromosomes, nucleus, nucleoplasm, nuclear membrane, nuclear pores. So everything has nuclear. Why? Because all of these are the structure organelles of an organelle. Got it?
Yeah.
Chromatin is a mass of coiled threads. I told you when all of these threads are coiled together tightly, they become chromatin. But if these threads are seen to be collected together, they form chromosomes. Like, look at this. They are just loosely coiled. So chromatin and these chromosomes basically contain DNA, and DNA is the deoxyribonucleic acid. And can you tell me what is RNA?
Only has one backbone.
Right, it has only one strand. DNA has a double strand. Exactly. Inside the nucleus, even more deeply staining area is the nucleolus. Got it? So this was all for your nucleus. Let's only do cytoplasm, and then I'll see if I have to stop. Otherwise, it's going to be too much for then. Then we'll do some questions from the end of chapter as well. You have your copy and pen with you or tab or something?
Yeah.
Good. All the living material inside the cell is called protoplasm. What is protoplasm? It's a mixture solution of enzymes, chemicals, hormones, neurotransmitters, substances, catalysts. They're all a mixture, which is called as a protoplasm. And protoplasm is present inside the vacuole. And vacuole's membrane is known as tonoplast. So remember, there are going to be a lot of terms in AS biology. And whatever I'm going to teach today, if you have to memorize it, you have to revise it today as well. Okay, we have classes of biology when? Wednesday and Thursday, right? So today and tomorrow we'll have classes of biology. And the same day you have to revise it as well. Otherwise, it's gonna become a very, very huge problem. Do you have any other classes after me as well?
Oh,
Okay. And are you taking other subjects academy as well? Marine biology and maths?
I'm taking math, marine science, not yet.
Hmm.
I'll only do it once my college done.
Remember, A levels best tip to study. The best study tip is that whatever you study today, like what have we done today? We have done the first three topics from your chapter one, including the microscope, the cell theory, and the organelles, plant, animal cells, basics of eukaryote, prokaryotes. You have to revise it today. If you want to make your notes, you can make those notes, but I don't recommend anything else other than the book for biology. All the other notes I'll be providing you. Handwritten notes I'll be providing you. You have to revise it. That's all you have to do. If you've done the revision, I won't have to give any other extra work over, because revision for biology and the day on the day of the lecture is very important. You don't use the weekend or anything else. Do it on the day I have teach. Okay, you can work at night after dinner. Just take the book, read it. Takes hardly 15 to 20 minutes. Got it?
Okay.
Yep. So it is also useful to have a term for all the living material outside the nucleus. We call it cytoplasm. Therefore, the cytoplasm plus nucleus makes the protoplasm. Protoplasm is everything inside the cells. It contains cytoplasm as well, and cytoplasm contains all the organelles as well. In total, whatever is present inside the cell is known as protoplasm. All organelles, the whole cytoplasm, the whole nucleus, everything. Cytoplasm is an aqueous watery material varying from a fluid to a jelly-like consistency, and using a light microscope, many small structures can be seen within it. These have been likened to small organs. We call these organelles, which keep swimming around this the jelly-like consistency, which is known as cytoplasm. An organelle can be defined as important definition. I guess it was mentioned above. I don't know. Victim. Okay. Fair. So organelle can be defined as a functionally and structurally distinct part of a cell. Organelles are often but not always surrounded by one or two membranes, so their activities can be separated by the surrounding cytoplasm. Each chloroplast, nucleus, mitochondria, Golgi, they are all separated by what? Cytoplasm. Organizing the cell activities in separate compartments is also essential for a structure as complex as an animal or plant cell to work efficiently as well. Next, if we talk about the cell surface membrane, cell surface membrane is present in all cells, whether it's a eukaryote, whether it's a prokaryote, it is all of them are surrounded by a cell surface membrane. We also call it plasma membrane. As mentioned before, it is partially permeable and controls the exchange of material between the cells and its environment. Nucleus we have already studied, so we have today done three organelles. What are those three organelles that we have done today? Cell membrane, nucleus, and cytoplasm. Are all these three clear?
Yeah.
Good. Let me see what questions can we solve from the end of chapter, and let's start those then. Cells, flagella, vacuoles, bacteria, comparison, viruses. Okay. Which one of the following? Nope. A to J. Okay. Let me give you some questions over here. Whatever the questions I'm going to give, these are going to be your notes. So whatever you write, write it very neatly. Use different colored pens. Are you going to write it on a tablet or a copy or what?
I'll write it in a book.
Good, on a book, okay? And you have to write it very neatly because these are your final end exam notes. These are your notes. Whatever the questions I'll give, it will cover everything that we have done today. So it becomes very, very easy. Why is it not opening?